Hi, we are team number 5, and we are Roberto Garzalaña, Daniel Alejandro Niño González, Manuel Maximiliano Gracia Lu, Mauricio Villarreal Rodríguez, Rafael Alejandro Pequeña Cuña, and Silvio Collar Chavez. And we start the topic with
The cell. Hi, I'm going to tell you about the cell. The cell is the basic structural and functional unit of all living beings. Every cell, from the simplest to the most complex, works as a miniature autonomous molecular factory. All the necessary activities to keep it alive are carried out frenetically and tirelessly. Some characteristics of the cells are that cells are complex and highly organized. Also, cells possess the ability to replicate. Cells replicate by division, a process in which the contents of a more cell are divided between two. Also, cells obtain energy in order to sustain themselves. And finally, cells interact with their environment. Some basic functions of the cell are that they are independent units consistent of a membrane. The membrane's function is to surround the cell and acts as a border or boundary with the outside environment. Another basic feature of the cell is that all cells contain cytoplasm. Cytoplasm is a membrane that encloses cytoplasm, which is a mixture of a jelly-like fluid and a variety of structures called organelles that are suspended in it. And the last basic structural feature of the cells is that all cells contain a medium life with genetic material that is called DNA. Uh, there are two types of cells that are the prokaryotic and the eukaryotic. Prokaryotic cells were the first organisms on the planet and are very abundant nowadays. The most important characteristic of these cells are that they lack a nucleus, contrary to their eukaryotic cells that they do have a nucleus. Thank you.
Hi, my name is Daniel. Now I'll talk to you about the prokaryotics, eukaryotic cells, the difference between them, and some characteristics of them. And also, I'll talk to you about the characteristics of a living thing and the most important things of them. Well, I will start with the difference between these cells, which are the prokaryotic and eukaryotic cells. Well, the prokaryotic cells are mainly known as very simple things because we have in here in the cell many absences, which they are the following: the absence of nucleus, the absence of membrane bond. These are unicellular and they don't have the vesicles nor vacuoles. And an example that we can find here, like the main example, are the bacteria. Now, continuing with the eukaryotic cell, are the opposite of these last cells, because in here we can find many more things, or just the opposite of the last ones. In here we can find a nucleus, we can find a membrane bond, the vesicles, the vacuoles, and the most important examples in here, where the most common ones are animals, plants, etc. Now, continuing with the characteristics of living things, we have that they can move, they can breathe, they can reproduce, grow, and the most important are that they interact and they respond to the stimuli, which it means when we as humans, for example, we make contact with fire, we get burned, like probably our hand, and we move faster in order to take off our hand, and also that the living things can adapt easily to the environment. And that's all. Thank you.
Okay, well, so now I'm talking about the history of the microscope and cell theory. And well, okay, in the first century, we obviously we can't see things with zoom, or we only can see things that our eye can see it. So it was a lot of time like that, but in the next few centuries, like from the 16th century, it was possible to see small spherical fragments while using something like a lens. These lens were used for constructing the microscope. And well, yes, in the 16th century, as I mentioned, a Dutch father and some pairs named Hans and Zacharias Janssen made the big compound the big compound microscope that was the microscope with more than one lens, where they place one lens at the top of of one cylinder and another at the bottom of the tube. And while looking at this cylinder, you can see 3 to or 9 times more bigger the object. In 1665, cities did not yet have the cleanest and measure where like we have right now. So there were a lot of unhealthy situations that were daily occur because they do not uh, well, they do not clean all the microscope because they don't know how it could take some bad bacteria or something like that. Today we know that Hugh observed the cell walls surrounding lifeless plants in the crack. Sorry. And while Robert Hooke was pioneering in microscope in 1667, a Dutchman in Delft who was making a living selling cloth made fascinating discoveries of his own. The Dutchman was a master of making spherical lenses of exceptional quality, which he placed between two metal plates fitted with adjustable screws that allow him to focus specimen. This design perfect the simple microscope and allowed him to magnify objects 200 to 300 times their original size. In the microscope, we have a mechanical system and an optical system that it depends on a lot of parts and objects. And in the cell theory, is basically that all living things are made up of cells. Therefore, they are the morphological and structural unit of all organisms. It is the physiological unit of every living being. All cells come from another pre-existing cell, and the cells contain hereditary material, that is the DNA.
The levels of organization, all body functions such as maintaining healthy glucose levels, proper temperature, blood oxygen levels. The levels of organization function like a set of matryoshkas. The levels that make up life, we could start with a microscopic level, such as the atom. A set of atoms properly intertwined would form a molecule. For example, depending on its size, a protein could be made of hundreds, thousands, or millions of carbon, hydrogen, oxygen, and nitrogen atoms. The group of tens of billions of cells with the similar functions and structures of performing a particular task is known as a tissue. and the group of organs comprises systems such as the digestive, respiratory, urinary, skeletal, muscular, etc. Next page. The homeostasis, one of the main characteristics distinguish living from a non-living things is the ability to maintain the internal conditions despite the condition of the external environment is called homeostasis. It's a physiological system of process constantly measuring and compensating for changes in the outside world that could damage internal systems. In the time of the ancient Greeks, Hippocrates' revolution medical concept to such an extent that today medical graduates also promoted that the disease because that diseases did not happen because of supernatural causes or divine punishment. Moreover, was the first to propose the concept of the body balance in a very rudimentary way, and in 1929, that the physiologist Walter Cannon named homeostasis the action of the regulatory mechanism, proposed every psychological variable should have a homeostat and negative feedback system.
Then it comes the cell transport. There are two types of cell transport. One is the active transport and the other is the passive transport. Um, Next page. The passive transport is made with diffusion. The diffusion is a process which molecules move to a higher concentration to an area of a lower concentration. There are three types of diffusion. The classic diffusion, and there is the osmosis, that is the diffusion of water to a semi-permeable membrane that separates two solutions with different concentrations of solutes. And then is the facilitated dissemination. If the substance that needs to across the membrane are ions or polar molecules of considerable size and low solubility, they must use a specific transport proteins. Then is the active transport. The active transport is basically the same as the passive transport, but requires energy. Um, Another name to call the active transport is endocytosis. In endocytosis, there are three types of endocytosis: the pinocytosis, the receptor-mediated endocytosis, and the pygocytosis. In pinocytosis, the plasma membrane forms a dimple that encloses contents outside of the cell and subsequently strangles at the edges, releasing small pockets of vesicles into the cytosol. The pinocytosis is the same. No, the pygocytosis is the same, but with larger molecules, and the receptor-mediated endocytosis involves blending molecules from the cell's environment, molecules of the membrane, which falls into a well and engulfs it.
Obesity in Mexico is currently considered a serious health problem, since 7 out of the very 10 adults live with excess weight problems. Some 300,000 minors, 36% of the population in basic education in Nuevo León, suffer from childhood obesity. Mexico has the highest adolescent birth rate of all the member countries of the Organization for Economic Cooperation and Development, with 62 out of every 1,000 pregnancies occurring in girls and adolescents, while the rate declined by 8 percent between 2000 and 2006. It went on to rise by almost 15% between 2007 and 2012. So here's the presentation about the Team 5, and thanks.